All right. Thanks, Neela. Well, good morning, everyone. It's an absolute pleasure to be here with you all. Hope everyone's enjoying a nice lunch today. Um, my name is Nicholas Plants. I have the privilege of leading the Washington Department of Fish and Wildlife's invasive mussel monitoring efforts. Today, I'm excited to talk to you about what we're doing to protect our beautiful state from these aquatic invaders through early detection monitoring. So just to give a little background on invasive mussels. When we're talking about invasive mussels today, we'll be referring to quagga and zebra mussels. Quagga and zebra mussels are freshwater mussels that have a unique anatomical feature called byssal threads. These byssal threads essentially act as sticky anchors that allows them to attach to hard surfaces. For any of you folks who work in the marine environment or may have grown up on the coast, you might know marine mussels that have these byssal threads, but there are absolutely no native freshwater mussels in Washington with the ability to attach to hard substrates. So, kind of a red flag for anyone working in freshwater environments. If you do see a freshwater mussel that's attached to a hard substrate or any substrate for that matter, that's a really big red flag. And so we just ask that you notify the AIS unit immediately. Um, Also, these invasive mussels reproduce rapidly. They have a high succession rate, resulting in densities greater than 20,000 per square foot, which overtakes aquatic habitats and critical infrastructure and can threaten our way of life here in Washington. They have multiple life stages, with the larval stage being microscopic and free floating, which makes them really difficult to detect at this life stage. But they're also able to disperse naturally downstream through water currents. And for adults, they're often much smaller than native freshwater mussel species, coming in at about an inch or less, about the size of your fingernail. And so I said they're much smaller than our native freshwater mussel species, which again lack those byssal threads. Invasive mussels were first introduced to North America through contaminated ballast water in the Great Lakes in the 1980s. And since then, they've spread from infested areas downstream and overland through movement of contaminated watercraft and aquatic equipment. And while we're fortunate they're not established here in Washington state yet, recent detections in the Snake River up in Idaho have put us on high alert. And we'll get into that a little bit more later on. So, why all the fuss over these mussels? Um, well, they're a serious threat to Washington's economy. We're looking at potential management and mitigation costs exceeding $100 million annually just for our hydroelectric systems. But that's not all. If we think about our fisheries, irrigation systems, drinking water, data centers, and even navigational locks, these costs just keep adding up. So these rising costs means higher production, logistics, and maintenance expenses, putting billions of dollars at risk in our state. The Columbia River, which is a vital trade gateway, handles a staggering $31.2 billion annually. It's our leading wheat export pathway and second for soy and corn. So if mussels take hold, shipping costs will skyrocket, affecting food transport and exports. Plus, 75% of our agriculture relies on irrigation, which is a $9.6 billion industry. Clogged intakes from the mussels could lead to lower yields and threaten food security. And let's not forget about the $20.5 billion generated by outdoor recreation and fisheries in Washington. Probably what most of you work at WDFW for. These mussels could seriously impact our beloved waters, jeopardizing $61.3 billion in economic activity just in these three areas alone. So if we move into the environmental risks, these mussels are notorious for destroying aquatic habitats. Because this infamous invader has been established in the U.S. since 1980, and since the 80s, there's a huge amount of research that can give us an idea of what we can expect if these mussels were to invade Washington. First off, they filter water at an incredible rate, where an individual mussel can filter up to a liter of water per day. So just imagine what a colony of 20,000 per square foot can do. They essentially strip the water of phytoplankton and zooplankton, which throws the entire ecosystem out of balance. And when we lose our native filtering species like native mussels and zooplankton, this leads to water pollution and decreased water quality. The water also becomes clear, which leads to increased vegetation growth and temperature changes. And we also see reduced oxygen levels in the bioaccumulation of pollutants, which harms native species like steelhead and salmon, which can even have indirect impacts on the recovery of our southern resident killer whales. Unfortunately, it doesn't end there, though. Research shows that these mussels can lead to botulism outbreaks, which is a dangerous neurotoxin. And as we know, pollutants and toxins can climb up the trophic food web, eventually reaching us. Invasive mussels also increase levels of harmful cyanobacteria, which can increase the frequency of harmful algal blooms. But we also got to consider the cultural impact. These mussels threaten Washington's identity, threat treaty protected resources, and the many benefits we receive from our water resources. The cost of mitigation and recovery will unfortunately fall on everyone and could mean losing the places we love. So if we take a quick look at this picture up here in the top right, this is a picture of a beach somewhere in the Midwest that's been infested from a water body that's infested with invasive mussels. And because these mussels produce such large populations, once they die off, their shells have nowhere to go but wash up on the beaches, and it essentially makes these beaches unusable. Growing up in the Midwest, I remember many times as a kid going out to our local lakes, and you couldn't even walk on the beach without shoes. You just cut up your feet. And even going swimming, these mussels attach to plants. So no one likes swimming in, in the weeds, but even more when your legs are getting cut up from these sharp shells that are attached to plants in the water. So when I say, we could lose the places we love, I'm really not being dramatic about that. It really has a cultural impact too to Washington. So again, I want to reiterate that there are no known populations of invasive mussels in Washington state. The closest detection in Washington, which many of you may have heard of, was in 2023, which occurred at Twin Falls in Idaho on the Snake River, which is an artery to the Columbia River. The detection is 375 river miles from the Washington border, which is a distance that water can travel in just a week. So this means microscopic larvae using water currents could travel to new areas and could very well be within our border. And that's what makes our monitoring efforts all the more vital. I also want to mention too, so the AIS prevention unit, we have a couple different ways of stopping the spread of invasive mussels. So I lead the early detection monitoring, but we also have watercraft inspection stations. And most commonly, when we discover boats coming into Washington state that are fouled with invasive mussels, we find that they're transporting their vessels from the Great Lakes and have Washington state or British Columbia as their destination. But we also know there's a lot of risk when people travel south for the winter to places like Lake Mead and Lake Havasu and return to their homes for the spring or summer. Lake Mead and Lake Havasu, as you can see in this sorry, laser pointer. In this picture here, these are most areas of concern because they're closest to us right now. But going back to the Snake River, despite media action and treatment application, quagga mussels were discovered again in the same area in 2024. So in October 2024, Idaho treated a 6-mile section of the Snake River for 10 days with chelated copper as a part of a multifaceted plan to eliminate mussels. Now, this is one of the largest treatments in the United States, but only time will tell if anti-mussel efforts are successful. As the previous elimination efforts have failed, we can't be certain that the latest round of treatments will eliminate all invasive mussels from the Snake River, so that puts Washington in jeopardy. Now, this brings us to the efforts that my team and I are doing to monitor for invasive mussels in Washington state. So what is early detection and why is it important? Early detection is exactly how it sounds. We have multiple sampling methods that we use to essentially look for a needle in the haystack, and we're hoping to find it as soon as possible. This is important because the sooner we can detect the species, the better chance we have at eradicating it, or at least mitigating the negative impacts it may have on Washington's waters. This graph here, I really love this graph. It illustrates the invasion curve. So it's showing the area of occupation as a factor of time. And the simplest way to put it is the longer we wait, the more difficult and costly it becomes to manage these invasive species. So that's why I consider our teams essentially on the front lines of preventing devastation that we've seen in other states. First and foremost, we at WDFW can't do it alone. I work with many partners across the state, ranging from federal, municipal, tribal, and universities, where communication is key between us to ensure we're doing the most effective and most efficient monitoring we can in the state. We have a little saying we like to use, and that's we're the best in the West, and I truly believe that. We have a lot of water in the state, as you know, and a lot of services we receive from these water resources. So the more collaboration we have, the better. With all this water in mind, we have to determine which water bodies are most at risk. This is calculated by numerous factors, including water chemistry traits, number of public boat ramps, accessibility, fish stocking, fish tournaments, public infrastructure, and more. The water bodies get a total score dependent on these factors, and we rank them as high risk, medium risk, and low risk. And then the number of times we sample a site depends on this categorization, where high risk sites get sampled 3 times a year, medium risk sites get sampled 2 times a year, and our low risk sites get sampled 1 time a year, or 1 time a year. And we just finished an updated water body risk assessment, which will assess over 900 water bodies and include updated data from 2020 to 2024. As you can imagine, it's a pretty massive team process to accurately collect all this information for over 900 water bodies. So the fact that we got that done, it feels pretty good. So just to give everyone a quick screenshot of all of our monitoring sites, this is from last year. So this is kind of the area we coverage. Sites are predominantly located at boat ramps or marinas, and they're all throughout the state. So we have 4 teams stationed across the state. We have one in Olympia, one in Spokane, one in Ephrata, one in Pasco, and they all specific in their own specific regions, or all sample in their own specific regions. So if you're at one of these offices, you see a tan guy like Mike out here, feel free to say hi. We're also looking at adding a new team down here in Region 5, which is where I'm at in Ridgefield, where they will be primarily our lower Columbia River crew, and it's going to really help expand our monitoring capabilities. So our sampling methods, we use a variety of sampling methods, including plankton tows, artificial substrates, shoreline surveys, water quality tests, eDNA sampling, and more. We're even looking to use plant rakes and ROVs soon, as well as collecting water samples at desired depths with a Van Dorn sampler. Each of these methods, these new methods that we're looking at adding, will help improve our effectiveness of monitoring in a few different ways. So the ROV can be used for image-based surveys to identify adult mussels attached to substrates, whereas right now we're very limited to only using human divers to go under the water and essentially, again, look for a needle in a haystack. So having a robotic ROV that can do the same as a diver and even get into places where divers might not be able to, like around hydropower infrastructure. So it just gives us a lot more capability. We're also looking at adding the plant rake for mussels that are attached to aquatic vegetation. As I mentioned earlier, the mussels love attaching to weeds as well. And so we can put a plant rake in the water, collect some more aquatic vegetation, and get some more samples under the water that way. And then the vertical water sampler, that'll help us collect water for our eDNA and our calcium samples in the middle of the photic zone rather than from the surface. So we might have a better chance at identifying either eDNA or collecting a better accurate calcium sample. Now I'm going to go into kind of our sampling methods. One of our main ones is taking plankton tows. We do this to look for juveniles or veligers using a 64 micron net. We do this once water temperatures hit above 9 degrees Celsius, as this is the minimum temperature for spawning, and when veligers will be free floating in the system. We do a horizontal and vertical tow at each site every visit, and these samples are then analyzed by private consultants in Florida and New York. We also use artificial substrates or collection plates for post-settled juveniles and adults, which are left in all year round and monitored by our AIS field staff. So just to give you an idea, these mussels, they spawn through broadcast spawning, meaning both the female and the male essentially shoot their gametes into the system and hope the best for fertilization. They do have a fairly high succession rate for fertilization, which makes them so invasive. But once these mussels are fertilized, or the individuals are fertilized, the embryo will develop into a free-floating veliger, where it filter feeds and grows into a juvenile veliger. As the shell develops, these veligers get heavier and heavier, and eventually they'll settle on a preferred substrate where they grow to become an adult. So we put these substrates up at our boat ramps and even near ports, as these are our most likely vectors for introduction if they're transported into the state. So the idea is that if they are transported into an area, the next generations of settling adults will be within the vicinity of our collection plates. We also do visual and tactical shoreline surveys, where we look for post-settled juveniles, adults, As I said, we have two-person teams, so one person will go left of the dock, or the other person will go right of the dock, and we basically do a 10-minute look down, pat down, looking for any settled adults, any husks of shells, dead adults that may have washed up on the shore. And basically, we're just looking and feeling around for any no mussels. So you can feel mussels as they're develop in this post-settled juvenile stage. It'll feel kind of like sandpaper when you're rubbing your fingers across the infrastructure, the dock in this case. So we also do a lot of water quality assessments, but a major parameter that we use in our risk assessment is the dissolved calcium concentration at each site. Dissolved calcium is a key environmental factor that limits the successful establishment of invasive mussels. So we've been taking dissolved calcium samples for over 16 years as of last week, actually. I just checked, our first sample was April 23rd, 2009. So we have a pretty good collection of calcium data for all the water bodies in this state. Excuse me. And just to go into more detail, invasive mussels, they require a certain level of dissolved calcium in the system to develop their shell as they grow. So without enough dissolved calcium in the system, essentially the mussels could be transported into a water body, but they won't likely establish because they won't get to that stage of becoming an adult. Now, other water quality parameters that can determine invasive mussel establishment, the probability of that, are salinity and pH, but we also take temperature and visibility measurements with a Secchi disc. This becomes important in determining the photic zone, which we use for determining the depth of our horizontal plankton toe. Because the planktonic veligers are filter feeding phytoplankton, they're most likely going to be found in that photic zone. So at this point, I'm sure most of you are familiar with eDNA. I know it's a bit of a new tool, but I think it's taken hold really quickly. So I won't go into too deep on this particular method, but we started taking eDNA samples in 2017 for dreissenid mussels only. So that's the genus that zebra and quagga mussels are in. So essentially, if we were to analyze a sample, we would get confirmed if there was a positive detection. We would only be able to know that it's in the dreissenid genus. But then we developed a few more assays in 2018 and 2019, including New Zealand mud snails, which is another aquatic invasive species, and northern pike as well. But then 2024 and 2025, we really started making our strides when in 2024, we were able to develop a species level assay for both zebra and quagga mussels. So now we can genetically confirm the identity of species, whether it's an adult or a veliger, rather than just visually confirming it's that species. And then 2025, we added a new invader to the United States, the golden mussel. Golden mussels are another invasive mussel that's not related to zebra quagga mussels, but they've shown to cause the same devastating impacts in the areas that they've invaded. They were first detected in the United States and California in 2024. So that raised immediate alarms for AIS managers across the states. And we're really proud that we were able to get an assay developed for the gold mussels. So now that we can monitor for these mussels as well. And we use a Smith & Root backpack sampler. You can see here is one of our technicians, Dalton, out there taking a sample right now. Basically, at the end of that pole there that he has is a filter housing. And this contraption is sucking up water samples. So the water will go through the filter, and then that filter is where we analyze the DNA. We also take petite ponar grab samplers. So we use this when we're looking for post-settled adults along the benthos area. I like to call it the claw. The claw gets dropped down to the bottom of the river or the lake bed, takes a big scoop of whatever's at the benthic level, rocks, gravel, mud, whatever the substrate is. And then it's brought to the surface, ran through a screen, and then we look for any invasive signs of invasive mussels there. Again, whether it's a live adult or the shell of an adult. Last but not least, we also deploy AIS signage at each boat ramp. And I really love AIS management because we don't work with just those within our agency and our partners, but We also have to maintain a close relationship with the public, and doing this helps kind of promote good behaviors for preventing the spread of AIS. So on our YouTube playlist, we have training videos for how to conduct each of these sampling methods for anyone that's interested in learning some more. Feel free to go check it out. So now this brings us back to the Snake River and how we at WDFW have responded to the detection in Idaho. Immediately after the alert in Idaho, two-person teams on a boat were sent to the Snake River, and we were there sampling within 2 hours. Since then, we've had a team on the Snake River, um, at least one team on the Snake River each week, and all of our samples from the Snake River get shipped overnight for a quick analysis. So it's conducted as soon as possible, and we get the results back. And since my short time in this position, we've been throwing quite a few curveballs, but through the storm, we were able to increase monitoring efforts in 2024 with a 6% increase in water bodies sampled, a 12% increase in sites sampled, and 13% increase in total samples. So this is, coming off the detection in Idaho, just making sure, again, we're really going through with a fine comb and getting as many samples as we can. And I really couldn't be more proud of my teams and have to give them a huge shout out for all their hard work and dedication over the past year. Now, I want to take a quick moment to talk about a new invader that I mentioned, the gold mussels. So these mussels were recently detected in California, the first detection in the United States. And though they're not related to zebra and quagga mussels, they pose the exact same risks, but perhaps even more worrisome is that their environmental restrictions or tolerance are a lot less than that of zebra or quagga mussels. Essentially, this means they can invade more water bodies than the zebra and quagga mussels could. This is due to their ability to withstand waters that have a lower dissolved calcium concentration, but they also have a wider tolerance to pH and low water quality. They can also withstand higher salinity concentrations. So when we're doing a risk assessment for these mussels, the amount of water bodies that they could invade or are high risk, it's almost essentially all of them. Well, Washington Administrative Code prohibits mussels in the same family as quagga and zebra mussels. As I mentioned, gold mussels are in a completely different family and therefore not prohibited in Washington State, despite posing risks similar to quagga and zebra mussels. Much like quagga and zebra mussels, gold mussels are believed to have arrived in the U.S. via international shipping activities, and determining the infested area is still ongoing, with new detections expanding the known infestation widely. As I mentioned, much like quagga and zebra mussels, gold mussels pose a serious threat to hydroelectric systems. Um, they were very well known to be invasive in Brazil. And so in 2018, they caused an estimated $120 million in impacts to Brazil's electricity sector. And so, again, just posing the same risks that zebra and quagga mussels do in the United States. So this detection in 2024 was pretty scary for everyone. And as I said, since their initial detections, gold mussel sightings have increased rapidly, indicating they spread without detection for some time, and highlights the need for consistent, widespread monitoring for AIS. We have a good reason to fear this mussel in domestic transport. For example, we discovered that there was this fishing tournament that kind of happens along the West Coast. And before they were coming into Washington to Potholes Reservoir, they were actually holding the fishing tournament in a delta. You can actually point here, right around here, coming out of California and going directly into Washington. So they would have been holding this tournament at a marina where they had a visual, a visual detection of an adult mussel back in December. So again, there's so many vectors and pathways for introduction, and it can be really tricky to stay on top of all of it. But it's just a reminder of why this early detection monitoring it's incredibly vital. That is all I have for you guys today. So if you have any questions about invasive mussels, early detection monitoring, or any of our sampling methods, yeah, please let me know and feel free to reach out.
Thank you so much. That was really interesting. Kind of scary, but interesting. Yes. Um, let's see. I have a question from Jonathan Core in the question and then if anybody else has any questions, feel free to write them in or feel free to raise your hand and then will you kind of mute yourself?
That's a great question, Jonathan. The really cheesy joke I like to use. My last name's Plants, but I am a fun guy and I work with mussels. So I'll leave the confusion to you guys. But yeah, it's a funny one.
I guess so. I'll kick it off with a question. What do you think the chances are that they're already here in Washington? We just haven't detected them yet.
Yeah, that's a great question, and probably one of my things that keeps me up at night. um, Absolutely, yeah. There's a high chance that they could be in Washington State. As I mentioned, the first detection in 2023 is only 375 river miles, or the distance that water can travel in a week. So, they while we haven't discovered any yet, there's a high probability that this year will be the year, but yeah, hard to say.
No, are there, oh, here we go. Well, actually, I'll ask my question first, and then we'll turn, then Laura can go. But first, are there other, you mentioned one mussel, a couple other mussels, but are there others coming down the pike that like you're looking ahead on that, that you read papers on that are also keeping you up at night?
Yeah, definitely. As of right now, zebra quagga and golden mussels are definitely our biggest concern. The golden mussel is kind of a new one, as I mentioned, first detection in October 2024. So a huge alarm for everyone, right? And I think it gives us good reason to look at any other species that could, you know, present the same risk that these three mussels do as well. But as far as our detection and monitoring, we kind of are just focused on zebra and quagga and gold mussels at this time. But like I said, we also do eDNA monitoring for the New Zealand mud snail, northern pike, alligator gar as well. So basically, if we can get these assays developed for aquatic invasive species, then each sample that we do take with eDNA, we can analyze for those species. So it's kind of, yeah, as soon as they are become a problem, right? And then we start thinking about it.
Okay, thanks. Laura, do you want to unmute yourself and go ahead?
Yes, can you all hear me?
Gotcha.
Wonderful. I had two questions. One, I understand the ongoing monitoring and that the goal of the monitoring is to not get anything exciting. But as you are monitoring, it sounded like it was like substrate and water quality and veg and all these other things. Are there any other data sets that you can contribute to with that to kind of answer some bigger, broader, different questions for other organizations or other groups? Is my first question, and I can ask the second, or do you want to answer that and then I can come back?
Yeah, quick answer to that. Yeah, so if anyone ever asks us for, again, like calcium data, salinity data, any water quality assessments too, we're very happy to provide that because, again, we've been taking calcium, for example, since 2009. So we have a really great collection of that data. And so it can certainly be used in other projects as well. Just anyone can feel free to ask. Yeah.
Oh, thank you. Yeah. And then my second question was, so you had said this kind of in the beginning where obviously the goal is eradication, but for zebra and quagga mussels, you also kind of then paused a little bit and said, or mitigation. So what does the mitigation of such an invasive species like that look like if it's if we can't eradicate it early on, is there a trajectory for mitigating and keeping them at bay?
Yeah, and so it's a great question too, and that's where all those economic costs really come into play, right? So if we look at the other states, especially the Midwest, you can see from the distribution map I showed, there is a great deal of quagga and zebra mussels on the eastern half of the United States. So we can use a lot of the information from them to see what we might experience if they were here. And a lot of that mitigation and maintenance of infrastructure, it all just comes down to very, very costly. So the estimates right now are over $100 million annually just for the hydroelectric systems alone. So that's where, if we are able to eradicate them, that's, obviously what our goal is. But there's not many examples of that being successful throughout the United States or even in the world. So then it really comes into the play of mitigating, managing, and there's a lot of technology they're trying to develop for anti-fouling. So that would actually prevent, for example, if you were to paint the infrastructure with an anti-fouling paint, you could prevent the mussels from attaching to that substrate. But obviously, anti-fouling paint has a lot of environmental side effects as well. So it's one of those things where it's just trying the best to reduce the impact as much as possible at that point. But again, it just becomes very, very costly. Of
Thank you.
course, thank you.
Any other questions? People welcome again to put them in the chat.
Yeah, that's a good question, Jonathan. I'm not particularly sure of any native freshwater mussel species in Washington that have invaded other areas. That's a good question. A lot of it comes down to, I call it kind of natural filters that prevent certain species from invading other areas. And really what makes invasive species unique compared to other species is their ability to tolerate wider ranges of environmental characteristics, and basically allows them to enter more environments than some other species would be able to.
And then, yeah, do you see David's question there?
Yes, yeah, read through it right now. Yeah, and we can analyze for native mussel species as well. But as far as collecting, like if we were to take a ponar grab and to find native mussel species, we don't record that information. But yeah, I agree. That would be a useful addition. Yeah, thank you, Rody. That's a great point. Exactly. So, like I said, a lot of these samples can be used for other projects and everything. So, really great range. And like I said, feel free to reach out to us at any time for any questions on if we could provide any data or information to anyone else. Looks like got most of the questions answered.
Someone asked, could you comment on their habitat range and how tolerant they are to brackish water?
Okay, perfect. Yeah. So zebra and quagga mussels, they have a lot lower tolerance to salinity. So in those brackish water environments, we don't monitor necessarily for zebra and quagga mussels. But now that we have the gold mussel, we're really increasing our monitoring efforts by, we actually have a team out right now collecting salinity levels at low tide and high tide in the lower Columbia system, so that we can get an accurate assessment of the risk in that water body. So, like I said, zebra and quagga mussels aren't able to withstand salinity as much, but gold mussels absolutely can invade the brackish water areas.
Can you tell us when you're looking at, so like if I go to, the ocean and I look at, I guess maybe you're talking about freshwater, but if I go to a dock, there's a lot of those black mussels. I thought they were local. I thought they were native.
Yep. Exactly. Yep. So we do have marine native mussel species that have byssal threads as well. I believe the blue mussel is one of them. I grew up in the Midwest, so I'm still learning my marine species, so bear with me. But yes, there are marine native mussel species that do have the byssal threads. So that's kind of like I said, if you're 100 miles inland, working on some lake or a river, and you find, a small mussel, like I said, about the size of your fingernail, and it's attached to something. And these byssal threads, the secretion, the glue that they use, has been looked at by NASA for aerospace engineering. It's a really strong secretion, right? So you'll know if it's stuck on there or not, right? And so, yeah, big red flag. Like I said, if you're in an inland freshwater body of water, if you do see a mussel attached to something, please let us know.
Great. There's an interesting post by Chase Gannell about kind of what could happen if we have to transition from invasive mussel mitigation to mitigation if eradication fails. So anyways, I guess that's a pretty interesting example though of why we all need to keep being careful, because you're in a better position now than we are with the green crabs.
Yeah, exactly. And like I said, we can look to the other states in the eastern United States and really see what's happened there, and kind of use that as a baseline for what we can imagine here. And none of it is good. It's a major issue here in Washington State solely because of how much we rely on surface water for, again, irrigation for agriculture. I've talked to farmers, and if anyone knows any farmers, they are usually on a pretty tight line year to year financially, right? So any added cost to them, it just the bottom down or top down effects economically on everyone in the state. I think one of our technicians who's been here for a long time, Mike Wilkinson, some people might know him. He likes to say, like, if you flush your toilet, you're basically affected by these mussels. So yeah, it's pretty scary stuff.
Okay. I think, yeah. Okay, yeah, thanks, Brian, for that more information. If we don't have any more questions, in the zebra quagga natural habitat, what keeps them in balance?
Yeah, that's actually a really great question. So they're native to Eastern Europe, Eurasia kind of area, and they are kept in check by natural predators such as birds. Birds are their main predator in their native range. And so there's also fish species too that can digest these mussels. But here, most of our fish species, unfortunately, they can try to eat the mussels, but because their shells are so strong, they can't digest them. And so essentially they come out the other end, and they're still viable. So in their native habitat, they have natural predators that kind of keep them in check. Yeah, and that's great information. Thank you so much for that, Brian.
Yeah, so there's also a nice Medium blog post you can check out there. Thanks for posting that. And if we don't have any more questions, we'll let everyone get going, keep going with their Wednesday. And again, this talk will be posted in the next couple of days on the director's office intranet site, so others can watch it, or you can watch it again. So thank you so much again. Thanks to the Aquatic Invasive Species group for all you do, and we'll see you next week.
Thanks, everyone. Have a good one.